Very good morning to all the participants who are preparing for JRF, SRF, and also ARS net exam. So the topic given me is type of cultures, culture media, inoculation technique, isolation, maintenance, and preservation. This is a very basic topic. So everyone in microbiology, which starts from like culture, isolation, purification, and their preservation, maintenance. So the microbiology and pathologist has to work on this aspect. So I'm Dr. Samadhan Bagul, working here in Directorate of Medicinal Aromatic Plant Research, Anand. So I will start now. So what are the types of cultures? The types of cultures, you can see mixed culture, pure culture, or it is also called as axenic culture and synchronous culture. Mixed cultures are two or more than two types of organisms. They are mixed in a plate. So it is a mixed culture. While the pure culture, it is single type of organism which is free from foreign material or any other impurities. This is called as pure culture. Or like axenic, which is also same single type of organisms. So this you can see in this plate. These are four types of pure culture, or you can say axenic cultures. Like another is synchronous culture. Synchronous culture is like the most of the organisms are in the same stage of cell division. Like you can see here this curved line and a straight line. So this curved line means they are in same stage of their growth cycle. So it is called as synchronous culture. While asynchronous culture is that is means it is not in same stage of cell division. So culture media. Culture media are used to isolate and identify bacteria, reveal their metabolic properties, and allow long-term storage of pure cultures. There are four principal culture media are there, which is one is based on composition, that is defined media. There is predetermined composition as there of nutrients, which is exact composition is known. For example, TSA, Na, or hello, sir.
Yes, sir, you are audible.
Okay. Then complex media. The complex media is a mixture of different nutrients. Like you don't know the exact composition, like peptone, beef extract, and other peptone, beef extract, and other complex substances are there. So their composition is imprecise. So example is peptone water and nutrient agar also. Then other is selective media. Selective media is which is addition of specific substances which favors growth of few microorganisms, which is the McConkey agar, Lowstein Jensen's agar. These are the example of selective media. While another is differential media, that is selective. It can be selective or differential, that differentiates microorganisms. Example, McConkey agar, which is selective for gram-negative bacteria, and which can also differentiate ability of lactose fermentation of these microorganisms. Then another media for preservation and storage of culture is then storage media, which is required for long-term storage of bacteria. Example, egg saline, medium, and chopped cooked media broth. Then another media is transport media, which is for temporary storage of specimen to bring into the lab. Example, thioglycolate broth for anaerobes. Then anaerobic media, which is it requires reduced oxidation and reduced potential condition and extra nutrients. While boiling the medium can expel this oxygen, so which can make anaerobic in nature. So the example of this media is Robertson's broth for Clostridium. Then another is type of media which is on the basis of consistency. That is, you must know that solid media, liquid media, and semi-solid media. The solid media is defined when the agar concentration is in the range of 1.5 to 2%. So it is makes a solid media. Then liquid media. So liquid media is also called as broth culture, where no agar is added. So another is semi-solid media, where the agar concentration is less than 0.5%, which is called as semi-solid media. So these are the basic types of media. Then another topic which is in process is isolation. So what is isolation? In isolation, in microbiology terms, refers to the separation of strain from a natural and mixed population of living microbes present in the environment. So it can be from rhizosphere, it can from endophytes, root nodules, or any other water sample, or any niche of the environment. So we will see one by one. So collection of rhizosphere soil samples is required for the isolation of rhizosphere from the roots of the plants. So roots of the plants is required. So draw the rhizosphere soil from healthy plant in the field. Then sample should be taken from a minimum of 5 to 10 points along a diagonal transect from each field. Carefully dig out the plant and collect roots added to soil that will represent rhizosphere soil. Place collected rhizosphere soil sample in a polythene bags and transfer to the laboratory until further process. Do not keep the rhizosphere sample in plastic bags for a long time, as it will lead to loss of microorganisms. Grind the soil sample finely and sieve through to sieve. Take a portion of soil sample and determine the moisture percentage, nutrient composition, etc. as per standard protocol, as per requirement of study. So and then another is isolation from root nodule. So basically, the root nodule is required to isolate rhizobacteria, which is symbiotic in nature. So particularly in root rhizobium, then rhizobium group, which is present in these root nodules. So what you have to do is take the legume root with the nodules, then wash with water and detach the nodule. Then nodules with root is detached. Then it add into these petri plate 70% alcohol. You rinse it with 70% alcohol for 10 seconds. Then again it is rinsed with sterile water. Then again rinse with 0.5% sodium hypochlorite for 5 minutes. Then again rinse with sterile water. And to confirm the success of surface disinfection process, you have to plate this water also in plating, during plating. So you can see that if this no growth is there, that means you can sure that our sterilization is okay. So to streak this sample onto this yeast mannitol agar plate with addition with Congo red, so it will give you this rhizobia. So rhizobia appears as white, translucent, and glistening, and elevated colony. Then another isolation is endophyte. What is endophyte? Endophyte is microbes present in root, stem, flower, leaf, all the parts of the plant. That is inside plants. So it is called as endophyte. So sample collection and wash thoroughly under running water to remove dirt. So which is the external dirt is there or any other foreign particles are there, it should be removed. So so that the contamination is less. So root, stem, and twig, leaves, it should be washed with water first. Then the same procedure just we have saw earlier. You just add in sodium hypochlorite solution, then wash with ethanol, 70% or 90% ethanol, 2% sodium hypochlorite, then again sterile distilled water. So after washing this after washing with sterile distilled water, you have to cut these pieces of this plant material like leaves, root, stem. Then you have to place on this media, the respective media. The mixed population of this isolated microflora will be seen in this plate. So you have to pure these isolates, and based on molecular technique or morphological units, you have to give the identification of these microbes. This is in brief the isolation of endophyte, rhizosphere, and from the root nodule. So how to isolate this? Many of you know that how to isolate. So serial dilution. This serial dilution procedure, where you have to take 1 gram of soil, 9 ml of water, that mixes this concentration you can see here. After that, 1 ml of this sample you have to take and dilute it into the 9 ml of broth, or 9 ml of water, sterile water. Okay, so from this 1 ml of from this dilution, you have to take 1 ml and dilute it into the another 9 ml of water. So it gives 100 dilution, and making serially it gives dilution to 100,000, then 10,000, 1 lakh, likewise. So you have to make these dilutions and take the proper dilution for accordingly to take isolate this organism. So after plating on this, after plating, you can see here, like first dilution and second dilution, you have so many colonies. So it is too many to count. So you should avoid that. Because the error may occur. So you can see here 10 to the power 3, 10 to the power 4, and 10 to the power 5. You can see here less number of colonies. So it is better to go for 10 to the power 4 and 10 to the power 5. 10 to the power 6 is you can see here no colonies. Likewise, it may be there, but it is just a depiction. So accordingly, the colony forming unit per ml you can calculate the number of colonies into dilution factor divided by volume of culture plate. For example, here in this plate 285 colonies are there. So 285 into and the dilution factor is there 10 to the power 3 divided by volume of culture, that is 1 ml. So it comes to 2.85 into 10 to the power 5 colony forming units per ml. Now plating. The plating is there, two types of plating: pour plate method and spread plate method. So in pour plate method, you have to take 1 ml, generally 1 ml sample is taken, and in spread plate method, 0.1 ml sample is taken. So inoculate these empty plates, then inoculate this plate containing solid media. You have to, in pour plate, you have to just pour this media first, then inoculate the samples. While in spread plate, the inoculation is you have to take sample media, solidified media, then on that you have to streak the plate. Then split streaking, there are 4 types of streaking are there. One is curtain streaking, another is continuous streaking, third is radiant streaking, and another is T streaking, T type streaking. This is quadrant streaking. You have to start from here, then you have to go just streaking here, then again go to this Bunsen burner, and you have to just sterile it, then again start from this spot, then again sterile it, then again stop from this plot. You have to in this way you have to make these streaks. Then continuous streak, you start from this point, you can see here A, you can start this point A to just go for crisscross and go for up to the point B. Like this is continuous streak. And gradient streak, you have to just see here, go for from this point you have to just make and go towards end of the plate. And T streak is there, like T shape. You have to make this inoculation in T type shape. Then purification of culture. This is just one paper is published in 3 Biotech. This is our paper. So we have just developed this method for purification of this cyanobacteria. You can see here mixed algal growth is there in blue-green algae. So this mixture is diluted in slide slide and are viewed under microscope 10x, because purification of cyanobacteria is very difficult. So you have to go with this method, or you can isolate another method, but we found this approach is suitable. So you can see here. Then after observing this under 10 microscope, you have to take this inoculum into this tubes. So in culture media, that is a blue-green algae culture media BG11, and then microscope, after microscopy, transfer the culture into this flask. So the culture will be generated. Then after culture, so each tube will be having it is supposed to be each tube having one pure culture. So just check it with microscope and transfer into the culture suspension. So this any algal culture will be there. So you can also again view this under microscope. So this flask is containing only one type of organism, that is pure culture or axenic culture. So likewise, 4 flasks are there, 4 cultures there. So it will be 4 axenic cultures. This is called as mass D technique, microscope assisted slide dilution technique. So now comes to the culture collection. So in India, there are culture collections, that is 30 culture collection in India. However, in the world, there are total 712 culture collections are there. While there are total conservation of these culture collections includes 25 crore 49 lakh 969. This much amount of microorganisms are present all over the world. So these are a few examples of these culture collections in India. So you can see here this national agriculturally important microbial culture collection. This is the only one agricultural microbes, only one culture collection in India which have agriculturally important microorganisms. So which is located in Uttar Pradesh. Then another culture collections are there, like fungal culture collection is there. Then Vishwa Bharati culture collection is there. National Fungal Culture Collection India. National Collection of Industrial Microorganisms, that is located in Pune. Then National Collection of Dairy Cultures, that is located in Haryana. Then National Culture Collection of Pathogenic Fungus, it may be in Pune. Then Microbial Type Culture Collection, that is Chandigarh, MTech. MTech is the institute, CSR institute, and their culture collection is there, microbial type culture collection. Then likewise, your MPKV is there, biological nitrogen fixation project collection of agriculture. So this is the map of the world for where culture collections are present. So this data is recorded in World Data Center for Microorganisms. This is the apex body for culture collection regulations. So culture deposition, where culture, how culture is deposited. So there is a process in culture collections. First, you have to fill passport data form, where you have to give the information regarding source, from where you have collected these microorganisms. Then you have to give the information regarding temperature, at what temperature. Then media, in which media you have to grow, isolate this culture, or you are growing this culture in the media. Then pH of the media, then characteristic, like what is the functional characteristic of this microbes, why you are depositing, that is plant growth promoting, or this is pathogenic, or any other important characteristic. Then you have to give the morphology and genetic information also to fill the passport data form. Until and unless you don't fill passport data form, your culture will not be submitted. Then after filling this passport data form, the pure culture plate or slant should be prepared and sent to the culture collection. So after receiving in culture collection, this will be validated by the concerned curators for bacteria, fungus, and cyanobacteria. Likewise, after validation, they just validate by growing the cultures there in culture collection. If they found pure, then the further process will be starts, like preservation of cultures in suitable form, and then it assigns the accession number to the depositor by the culture collection. So now we come to the preservation methods used at NMCC. Sorry, this is from I have taken from NMCC, that is National Agriculturally Important Microbial Culture Collection. So this conservation of agriculturally important microbes, these are short-term and long-term methods. In short-term, they are used glycerol, mineral oil, while long-term, lyophilization and liquid nitrogen there. So you can see the images here, like this is a glycerol stock, there is a glycerol vials are there, and it has to be deposited in this minus 50 or minus 80 refrigeration, deep refrigerator. Then mineral oil is this mineral oil is there. You can just like paraffin oil, you have to just inoculate in this paraffin oil and store into the room temperature, or 20 to 25 degrees Celsius temperature. We will see further in this slide. Then lyophilization process. You can see this is the lyophilizer. It is a very useful process and widely accepted for storage of the microbes. Then cyanobacteria. For cyanobacteria, it is very difficult to store long term. Generally, this is stored in this live culture. You can see this is live cultures are there in this flask. This is lyophilization storage. This are it has to keep in ampoule and store in this room. Then liquid nitrogen storage. These are the cryogenic tank. From in this cryogenic tank, these samples are stored. Cultures are stored. What is the importance of preservation? To retain the authentic cultures. Then basic resource for research, like various students and various researchers require these cultures for their research. Then industrial use in bioproduction, like bioproduction, bioethanol production, any other like amylase industrial important enzyme production. So for that purpose, you can use these pure cultures, and that will get from this culture collection. Required in training program. It is also required for publication, where when you are going for publication, that publishers ask for your deposition number of this culture collections. So that's why it is so important from this preserved cultures. Then just we should short-term preservation. So, short-term preservation is subculturing. Subculturing, where serial transfer on agar slants or in liquid medium. You can go for subculturing and stock this culture at storage at 4 degrees Celsius. So this what is the disadvantages? There is contamination problem. Frequently you are transferring this culture, there is a chances of contamination. Then loss of genetic and phenotypic characters. Then induction of alteration in cultures. Then loss of culture. Then mislabeling of cultures, infestation with mites, and labor intensity. There are this kind of disadvantages are there while subculturing, because sometimes we handle it very poorly. That's why these problems occurs. However, another is that like frequently going for subculturing, there is loss of genetic and phenotypic characters. So another is this is subculturing strategy for bacterium. Minimal media are preferred as compared to rich media for subculturing, because the microbes grow in rich medium is faster. That is fastidious growth will be there, so that will be avoid rich medium. So minimal medium is preferred. So maintain for 2 to 3 months once refrigerated after subculturing. Then for fungus, subculturing period varies from fungus to fungus depending on nature of cultures. Storage in the refrigerator or cold room at 5 to 8 degrees Celsius can extend the transfer period to 4 to 6 months. The glycerol stock. Both bacteria and fungus are being preserved in glycerol stock. Generally, 10% glycerol is used for fungus and 20% for bacteria and 40% for actinomycetes. If you don't have the glycerol, then 5% dimethyl sulfoxide will also be used. These are the glycerol vials then used for this glycerol stock, that is storage of bacteria and fungus. Then preservation of fungus in mineral oil. You can see these tubes are there, and these tubes are filled with mineral oil. So this prolong the utility of stock cultures, that is 10 to 12 years. Exception maybe 30 years. This mineral oil prevent evaporation from this culture. Then decrease the metabolic rate of the culture by limiting the supply of oxygen. Then this method is more suitable than lyophilization for the preservation of non-sporulating strains. Advantage: storage temperature, that is 15 to 20 degrees Celsius, so that many labs can afford. So there is no high requirement of storage. Then best for mycelial or non-sporulating cultures. Then prevents from mite infestation. Then except for Zygomycotina, the storage of living fungal cultures under mineral oil was relatively successful. What are the disadvantages? It is not suitable for Basidiomycetes cultures. Fungus growing continuously, and the mutants can grow under adverse conditions. So there is also some factors responsible for this mineral oil preservation. You should not take so much oil, so it can prevent this oxygen level, and it should not be too low that it will dry out the culture. So it should be optimum. Then other preservation techniques: the storage over silica gel, that is Neurospora for Neurospora species. Then preservation on beds, the method involving preservation on beds, that is glass beds, porcelain beds developed by Liederberg. The preservation in sterile water, that is simple method. Another, it is very inexpensive method for storage of fungal cultures. In this preservation method, Basidiomycetes will survive for 2 years at 5 degrees Celsius. Then preservation of fungus on paper, which is developed by Rosalba Ruben and Ximena Ericapa. This technique is used by CIT, that is International Center for Tropical Agriculture, Colombia. It is used for preservation of insects, fungus, that is your bioagents, bioinsecticides, then plant pathogenic fungi and molds. Then another method is filter paper technique for preservation of spore forming fungus. That is, when this pure culture of fungus is there, you have to take this filter paper, sterile filter paper, then cut into the pieces and kept into the keep onto the this plate, sterile petri plate. Then inoculate this filter paper pieces with this mother culture or pure culture and incubate for 7 days. Then after incubating this filter paper with fungus, 15 to 20 days for keep for drying in incubator. Then after drying, this dried filter paper kept in packed in plastic bags. After packing in plastic bag, store at 4 degrees Celsius or minus 20 degrees Celsius. In long-term preservation, the preservation at low temperature, that is minus 196, minus 80 degrees Celsius. So this long-term preservations, which can be deep freezing at minus 20 degrees Celsius and minus 80 degrees Celsius. Then lyophilization, that is also called as freeze drying. Then cryopreservation, storage minus 196 degrees Celsius temperature. These are the some deep freezing that is routinely used in laboratories, where maintenance temperature is minus 5 degrees Celsius to minus 80 degrees Celsius, which stores for 1 to 2 years. Then another deep freezing method is minus 70 degrees Celsius to minus 196 degrees Celsius, that is 5 to 10 years. Then lyophilization, process of what is lyophilization? Just said that it is a freeze drying. It is a process of removal of water by sublimation under vacuum. That is the first step, primary step, primary drying. And then by desorption of this water, by desorption, this is secondary drying. The water passes directly from solid state to the vapor state without passing through the liquid state. You can see here, these are the ampoules for lyophilization, which requires for this storage. So these are the short brief methodology, brief process. The sample is prepared. Sample is prepared in skim milk. Then freezing is there. Then primary drying is there, where sublimation through sublimation occurs. Then secondary drying. Then final product is stored. What are the live protectants are used into the lyophilization process? Lye injury is a damage caused by the freeze drying and disrupts the fluid mosaic structure of the membrane. So to avoid this, you have to use lye protectants such as serum, skim milk, inositol, trehalose, peptone, which reduces the prospects of damage like freezing, dehydration, oxygen damage. There are many lye protectants. They are there. These are some examples. Just generally used, like skim milk is mostly used. Then advantages of lyophilization. Majority of the fungus, bacteria and yeast strains can be lyophilized. Then enhanced product stability in a dry state. Ampoules can be stored in room temperature. Shelf life of 20 to 40 years. Protecting agents help preventing from intracellular and extracellular crystallization. Disadvantages. This is not suitable for non-sporulating basidomycetes. Bacteria is sensitive to lyophilization. Involves examples: Aquaspirillum serpensis, Clostridium group, that is Clostridium botulinum, Chowai, Novai, then Putrefacium, Scatologens, and then Helicobacter pylori and Peptococcus reducens. Then Spirilla and Azomonas insignis are sensitive to initial freeze drying stage. Then liquid drying. It is used to avoid the damage caused by freezing. Then effective in preservation of anaerobes that are damaged by or fail to survive freezing. It is markedly superior to lyophilization for the preservation of unicellular algae, useful to culture which produces spores or other resting structures. Then cryopreservation. Cryopreservation involves storage of cells in liquid nitrogen at temperature up to minus 190 degrees Celsius. Then development of cryobiology. So how this cryopreservation is established? That is, first 1776, that is Spallanzani reported spermatozoa frozen in snow were able to recover their mobility after thawing. Then in 1940, Chang reported the birth of a litter of rabbits from embryos stored at 0 degrees Celsius. These two events is sufficient to establish this branch, that is cryobiology, where these microbes or any other substance can be stored at minus 90 degrees, minus 196 degrees Celsius. Then cryo injuries are there, and to cope up with this, the cryoprotectants you have to use. Concentration effect caused by this pH change, then precipitation of buffers, then dissolved gases are there, electrolyte concentration, intracellular crystallization. These are some problems are there which can be which makes cryo injury. So then cryoprotectants, such as polyols, which is glycerol, DMSO, that is dimethyl sulfoxide, then trehalose, sucrose, these are the sugars, then propylene glycol, this can be used as a cryoprotectant. So damage caused due to this freezing, where freezing technique is you have depends on the technique how you uses this freezing, like quick freezing, slow freezing, and thawing and recovering. This freezing in quick freezing, there is a formation of ice is there, so you have to prevent by adding protective agents. While slow freezing, the dehydration occurs, chances of dehydration occurs. So cope with pre-incubation at high osmotic pressure solution. While thawing and recovering, the cell is damaged, so rapid melting is required. So advantage of cryopreservation: length of storage is effective at minus 150 degrees Celsius. The majority of organisms survive well. Organisms remain free of contamination when stored in sealed ampoules. Almost negligible changes observed in morphology and genetics of microorganisms. Disadvantage of cryopreservation: high cost of liquid nitrogen apparatus and continuous supply. Then whole collection can be lost if cryo vessels are ruptured. Then liquid nitrogen contamination or problems with culture distribution occur often because there is handling errors will be there. Then accumulation of the damage caused by background radiation. When this cryo vials are start, there some vibration is there, some radiation comes, so it may cause damage. Then other than this regular microbes, there are recalcitrant microbes, which is prone to cryo injury. Organisms are prone to cryo injury or exhibit poor viability. Such organisms called as recalcitrant microbes, so which includes helicobacter, basidiomycota, pythium, promis, saprolegnia. These are the microbes. So specific protocol is required to store, to preserve these microbes. So preservation technology. In order to overcome the problems of preservation of recalcitrant microbes, new preservation technologies can be applied. That is vitrification technology, encapsulation technology, preservation using growth substrate. What is vitrification? Vitrification is an ultra rapid method of cryopreservation. So extremely fast cooling, that is 37 degree to 3196 degrees Celsius, is very fast cooling is required. Then high concentration of cryoprotectants. Then glass-like state is formed. Whole solution is solidified without the crystallization of ice. Common method to preserve, for example, human egg cells in oocytes cryopreservation and embryos in embryo cryopreservation. Then encapsulation technology. The cells are embedded in calcium alginate beds prior to cryopreservation. This you can see these beds are there. This is calcium alginate beds. Then water content of the cells may be reduced by osmotic treatment by drying, which prevents from damage caused by ice concentration effect during the cooling stage. Allow cells to easily handled and manipulated by providing a suspended matrix. Preservation using growth substrates, used in cases of obligate pathogens or mutualistic microorganism preservation. Then use of growth substrate or host has been applied for many years. Example, hemp seed are being used to support members of the promista when cryopreserved. Microcyclic rust fungus, that is Toxinia spoganzini, where the teliospore were present on the petiolous tissue. Alternative approach for many unculturable microorganisms. Some of the questions to be addressed in future research. It will be like which growth phase of culture is suitable for different preservation strategies. Then what is optimum number of initial cell density for preservation in order to get more viability. Then how would you design strategies to preserve polar region, hot spring, alkaline lake and marine ecosystem for preservation of specific microbes. How would you develop a cheap and less labor intensive protocol for existing microorganisms. How would you unveil molecular mechanism that induce morphological, physiological, and genetic changes during the course of preservation? How can we preserve microbial community in an ecosystem? Is there any variation in metabolic production before and after preservation? Then these are some observations or observations given by this DSMZ, that is German culture collection. Then this is BCCM CAPV. It recommends preservation of most microorganisms at -135 degrees Celsius and also storage temperature below -70 degrees Celsius. It is not acceptable. Prior to storage, the cells must be cooled at a rate and which production of intracellular ice crystal is avoided. Storage in liquid nitrogen, high survival rate, ability to preserve, growth morphology and production of metabolites. This is preferable above all. These are some observations is given by this culture collection for preservation in liquid and nitrogen. So with this I end this lecture. So I would like to acknowledge our former institute, that is National Bureau of Agricultural Important Microorganisms, Uttar Pradesh, and my current institute, that is Directorate of Medicinal and Aromatic Plant Research, Anand. And I also would like to thank Department of Extension, Mahatma Phule Krishi Vidyapith, for giving me this opportunity. So I hope I will provide this information more detail in written notes. So I hope if you have any questions, you can ask.
Dear participants, you may please unmute yourself and you can interact with the sir if you have any comments or questions.